let me introduce Milad Abolhasani. So he is an ALCOA Professor and University Faculty Scholar in the Department of Chemical and Biomolecular Engineering at North Carolina State University. He received his PhD from University of Toronto in 2014. At NC State University, he leads a diverse research group, the study of self-driving labs. He received the enormous amount of the awards, including NSF CAREER Award, IACL 35/35, Dreyfus Award for Machine Learning in the Chemical Science Engineering, and many others. So, Milad, stage is yours.
Great. Thank you, Alexei, for the kind introduction. And also thank the organizers for the invitation to talk about some of the work we've been doing in my lab over the past 7, 8 years to accelerate the way we think and approach materials and molecular discovery by integrating human knowledge, AI, and robotics. So we were looking for a new material or molecule. Most of the time, we are dealing with this infinite chemical universe. Unfortunately, we don't have a map of this universe. We have to know the target properties of the molecular material that we are looking for. We don't know how to navigate it. We don't know how to find it. We don't even know sometimes. We don't even know what that molecule is. So most of the time, we are just blindly searching through this space using a Dissonian trial error-based technique. So that's 50% of the reason why our materials and molecular discovery efforts are very slow and expensive. The other 50% of the problem is the way we navigate this infinite chemical universe. This is the way we conventionally approach experimental sciences. We send our highly trained scientists in the lab. They spend 70–80% of their time just doing repetitive tasks, maybe pipetting, preparing samples, running reactions, and characterizing 1, 10, 20 experiments a day. At the end of the day, our scientists go back in their office, look at the data, and use their intuition mainly to decide what should be the next experiment to run. So this is our conventional closed-loop science, but it's very slow and resource-intensive. Now imagine when we're dealing with high-dimensional problems, 10, 20 different parameters that we're dealing with. The approach that we typically take is one parameter at a time and one experiment at a time. So this is the other 50% of the problem that makes the materials and molecular discovery efforts are very, very expensive and slow. So looking at this picture, there's a lot of room for improvement. For example, we can leverage all the recent advancements in robotics and lab automation to bring our human scientists out of the lab, not out of the loop of experimental sciences, and then have this end-to-end automated experimental workflows to increase our throughput and reproducibility and reliability of our experiments, and then use recent advancements in machine learning and AI to better guide the decision-making process, better sample that infinite chemical universe, and have more informed sampling of that reaction space. So when you integrate that robotics lab automation with an AI agent, you create what we call a self-driving lab. Now, what you get out of this self-driving lab is that promise of that 100 to 1,000 time acceleration to conventional strategies. And we call this a robotic co-pilot, like the AI co-pilot that sits in the digital world. This robotic co-pilot bridges the gap between that digital and physical world. And by nature, these self-driving labs are more sustainable, so they reduce the carbon footprint of your discovery. And if you use scalable technologies in your synthesis and purification and scale-up processes, you can reduce that time from lab to market. So depending on how you integrate human knowledge with robotics and AI and automation, you can have different human AI robot collaborations in your experimental discovery workflow. You can start from piecewise operation all the way to self-motivated autonomous labs that we are not there yet. A self-motivated lab is where there's no human intervention and the AI agent comes up with its own questions and its own hypothesis and evolves over time. Where we are right now, and what I'm going to talk about today, is this closed-loop autonomous labs that the human scientist is still the driver, and the self-driving lab is the co-pilot that bridges the gap between the digital world and the physical world. So the digital world is your cheminformatics, materials informatics that you can do in silico screening and maybe optimization, but that information is still in the digital world. This robotic co-pilot or self-driving lab takes all that information, the target compound, maybe the synthetic route, and then tests that in a closed-loop format and verifies the hypothesis and maybe rejects it or comes up with a new hypothesis. And what you get at the end of the day, there's a lot of benefits of this robotic copilot, but the most important thing that you get is that reduced time to solution by 100 to 1,000 times, going from 10 plus years in your material molecular discovery down to a few months, ideally, and going from plus $100 million of R&D costs, hopefully less than $1 million for your discovery. And then within that robotic copilot, you can imagine having different modules, and each module, from your precursor formulation to synthesis, characterization, and testing, they can have their own AI agents. And then after this, the question is, how can I build an ideal autonomous lab? So this is an opportunity to rethink our experimental workflow. Instead of just doing what we have been doing for the past century, using flasks and having a lot of waste in the chemicals that we are using, this is an opportunity to think about material efficiency, chemical consumption, trying to re-engineer design of our automation platforms for materials and molecular discovery with this perspective of miniaturizing the chemical consumption so that we can minimize the waste generation of these autonomous operations. We have to think about precision and reproducibility of our automation workflows. Not every automation solution is precise. We have to make sure the data that we are generating through these automation technologies are precise and reproducible, because those are the data we're going to provide to that AI agent. The AI agent is trained on the external data you're generating in the lab, and if you have noisy data, then instead of accelerating your science, you end up decelerating your science. And we have to think about what are the most appropriate in situ online characterization techniques that provide the throughput that is needed to provide the data for that AI agent. And at the end of the day, what we want to have is that big, reliable experimental data. And then we have to think about how do we actually translate knowledge between different modalities of running reactions, different types of hardware, different robotics. You can run the same organic synthesis using a round bottom flask or a flow reactor or a parallelized miniaturized batch reactors. Now, depending on the reaction conditions or the environment of the reaction, you might actually get different results. Now, how do you augment these data? How do you transfer data from one lab to another lab? So understanding how we can actually come up with this language for different robotic labs that we can transfer knowledge and augment the data to further accelerate discovery is another important thing we have to consider when we are thinking about designing an autonomous lab. So thinking about the big experimental data, in my lab, instead of using batch reactors, which is the common gold standard to run different types of organic synthesis, we use flow reactors. A flow reactor is basically a microfluidic reactor. Microfluidic has been around for more than 30 years. But basically you're continuously flowing the precursors of your reactions, and the reaction is happening in a very small channel, and the reaction happens for a specific reaction time at a specific temperature, and that's the time that your reagents are spending in a very small microchannel that they are flowing. And one of the advantages of microfluidics is that enhanced heat and mass transfer rates and your photon flux absorption that you have. So in basically simple words, we can miniaturize our reactions. When we run these reactions in a microfluidic reactor, we can accelerate the kinetics of reactions. So if a reaction takes hours or days in a batch reactor and it's mass or heat transfer limited or photon flux limited, we can run the same reaction in a matter of minutes in a microfluidic reactor. So we can run hundreds of reactions at 3 or 4 orders of magnitude lower chemical consumption than a batch reactor. And then we can precisely automate the reaction, control all of the reaction conditions, and integrate different in situ or online characterization tools to characterize the compound that leaves the reactor. So these are some of the most advantages of microfluidics to provide that big experimental data that I was talking about in the previous slide for the AI agent of the self-driving labs. And we use 2 different types of flow reactors in my lab. One is a continuous multi-phase flow where we have a train of droplets that are flowing along the flow direction, and the reaction is happening inside these tiny droplets. The volume of these droplets in the order of maybe half a microliter up to 1 or 2 microliters. And we have another flow format that we call a single droplet oscillatory multi-phase flow, which in simple words, basically we have one liquid droplet or a liquid plug, and you're just oscillating this back and forth. It's like shaking a vial. And in this case, you can have a very small reactive footprint, let's say 40 microliter. We can have a droplet of 2 to 4 microliter and run reactions in the order of seconds, days, or hours, or maybe even months. So if you're dealing with relatively slow chemical reactions, we typically use this single droplet format. If you're dealing with relatively fast reactions or we want to do continuous manufacturing, we use the continuous multi-phase flow. So in my lab, we basically develop these bespoke flow reactors and use robotics and lab automation. We use liquid and solid handling robots to automatically prepare the stock solutions of our reactions, and then we develop our own bespoke online characterization tools, chromatography, spectroscopy, NMR, mass spec. And then we use an AI agent to drive the operation of this end-to-end experimental workflow. But what I want to highlight again here is that this self-driving lab is not replacing the human scientist in the lab. It is acting as a robotic co-pilot for that human scientist that is still providing the question and the initial hypothesis and even the constraint of the space that the self-driving lab has access to. And then we use this self-driving lab as a foundational technology in my lab and apply it to 3 different research areas. One is nanoscience, the other one is catalysis, and the third research trust, we use all the data and machine learning models that we develop in the nanoscience and catalysis areas to better understand the fundamental mechanisms of the reactions and processes happening in, for example, nanocrystal formation or transition metal catalysis reactions in the catalysis area that we are studying. So for this presentation, I'm mainly going to focus on the catalysis area. Here's an example video of how we can use robotics to accelerate our experimental workflow. So here is a custom-built robotic solution inside an inert environment. It's a triple glove box that we can automatically do solid and liquid dispensing, prepare the stock solution of our organic reactions, for example, CCC and cross-coupling reactions. We can heat mix our reagents, and we can even study the studiability of the starting precursors and start running the initial discrete screening of different parameters, different catalysts, different ligands, solvent, and base. And then we can basically select or find the hit conditions and then run those in our flow reactors for fine-tune optimization of the reaction conditions. And then once the synthesis is finished, we use mobile and stationary robotic arms to transfer samples to different stations for downstream processing. For example, if you want to do any sort of bioassays, we can do the transfer for the bioassay station, so we can have this 24/7 operation of our self-driving labs. But as I mentioned, we use flow reactors to run the synthesis, so we can do hundreds to thousands of different reactions per day. These are just 2 example videos of droplets in a very small flow reactor. So the diameter of the tube here is in the order of 750 microns. The volume of those blue droplets where the reaction can happen typically is in the order of half a microliter. And as I said, we can do up to 1,000 reactions, organic synthesis a day. So we have been working in this field of self-driving labs over the past 7-8 years, and here are some examples of these self-driving labs that we have developed both for inorganic materials and for organic synthesis. So as I said, I'm going to talk about the catalysis area today. So one of the most important or most challenging things that you always want to study when you're doing any sort of catalysis for organic transformations is how effective is the ligand that you're using in your organic synthesis, for example, Suzuki coupling, hydroformulations, or even CN coupling. So whenever you have a ligand, it's a homogeneous catalysis, for example, you want to know how effective that ligand is in controlling the selectivity and yield of the reaction. So typically, what chemists do, they have a list of conditions that they run. So they do this manual screening. So the white dots are the experimental data that are within the boundaries of the selectivity and yield or the best performance of this catalyst or ligand that you're testing. And the red dots are the Pareto optimal points that you cannot, for example, improve the yield or selectivity of this ligand beyond that specific point without compromising the other one. But what you ideally want to have is a method that can give you this very targeted selection of experiments that you can minimize your chemical consumption and in very short amount of time find this Pareto optimal points for a given ligand or catalyst so that you can accelerate the catalyst ligand discovery for organic synthesis. So I'm going to show you in the next few slides how we design a self-driving lab that can autonomously test the performance of a given ligand and then use that information to further accelerate discovery of new catalysts or ligands for organic synthesis. So one part of a self-driving lab is the hardware. And then I'm going to talk about how reaction miniaturization can accelerate throughput of reactions. Then the second part is the online characterization. And the third is your machine learning benchmarking, or the AI agent of your self-driving lab that can basically guide the selection of experiments and plan what should be the next experiment in order to intelligently navigate that chemical universe that I was talking about in the beginning of my slides. So in terms of reaction miniaturization, as I said, we use two different types of reactors, a single droplet or a continuous flow reactor. But these are basically miniaturized reactors that you can run reactions in the order of half a microliter or a few microliters. And then we can reduce the chemical consumption by 100 times, accelerate kinetics by 5 to 10 times, and run reactions up to 1,000 organic synthesis a day. We can easily pressurize reactions, go up to thousands of psi for the pressure, and run high pressure, high temperature reactions reliably, continuously in these platforms. And what we have done in the past, we've shown we can use these flow chemistry platforms to run a range of different organic synthesis, from reductive amination to Suzuki coupling, SNAR reactions, photoredox catalysis, and C-N cross-coupling reactions. They're just, this shows the diversity of different reactions, organic synthesis that you can do with these flow chemistry platforms. Here's an example of this fully automated end-to-end experimental platform using robotics for liquid handling, automatic precursor preparation on the left, and running the actual reaction in a 4 microliter droplet in the middle in that single droplet reactor, and then doing online analysis on the right with an online HPLC ELST mass spec to characterize the yield and selectivity of this reaction. And here's how you can use liquid handling for automatic precursor preparation, and then this is where you can run your reaction in a few microliter droplet. You can do both gas liquid or liquid reactions, single phase or multi-phase reactions. You can also couple this reactor with a collimated light source to do photoredox catalysis. Here's an example of doing a photocatalysis using semiconductor nanocrystals at a photocatalyst with different band gaps for organic transformation. And then once you optimize your reaction using autonomous experimentation, you can also scale up the synthesis, going from that single droplet to continuous flow. And the knowledge is directly transferable. It's a scalable knowledge that you discover, and you don't need to do any additional optimization. You can just directly transfer the ligand catalyst and the reaction condition to discover to a continuous flow reactor and continuously produce the target compound with the optimal conditions that you discovered in your single droplet reactor. So that was the hardware, the reaction miniaturization side of your self-driving labs. The second part is how you actually make a decision about what should be the next experiment to run. This is where we do a lot of in silico process optimization to make sure we have the most optimal fine-tuned AI agent for self-driving lab. So in this case, I'm showing a Bayesian optimization framework where we wanted to improve the Pareto optimal points, or the Pareto front of selectivity and yield of a specific chemical reaction, in this case, hydroformylation of olefins with different phosphine ligands to produce aldehydes, which are the starting chemicals for a lot of alcohols and other APIs. So in this case, basically we're using hypervolume improvement or expected hypervolume improvement, and you start your reaction with initialization of some either randomly selected experiments or design of experiment methods to initialize a surrogate model of your Bayesian optimization. Then you can model the future experiments and rank them based on their expected hypervolume improvement, which is basically, in simple terms, what is the probability of running a specific experiment in the future to improve the yield and selectivity of this reaction. And then we select the top rank experiments, send it to the hardware of our self-driving lab, automatically run that reaction, and update the surrogate model and run through this loop until we cannot further improve that Pareto optimal point. Now, in order to fine-tune the AI agent of the self-driving lab, we did a lot of in silico modeling here to make sure the brain of our self-driving lab is fine-tuned and it's the most optimal modeling and the decision-making on the uncertainty, so that we can find or map the performance of a given ligand in the shortest amount of time possible, so we can minimize the experimental cost of ligand and catalyst discovery. So I'm not going to go through all the details of results I'm showing here, but we looked at different the surrogate modeling architectures. We looked at different Pareto front decision-making policies, and we did more than 600,000 simulations to fine-tune the hyperparameter and metaparameters of these this AI agent of the self-driving lab. And after that, we integrated that fine-tuned AI agent with this fully automated experimental platform that starts with automatically preparing the stock solution of your ligands, catalyst, liquid handling, solid handling, and then we basically perform the reactions in a 0.5 milliliter scale volume. This is again a reaction of making aldehydes that you can use again to make different amines, different alcohols, and different APIs. And then the product was automatically analyzed online to update the surrogate model of the AI agent so that it could automatically select what should be the next experiment to autonomously map the performance of a specific ligand or catalyst given this reaction. So here's a picture of the actual experimental platform, different fluid delivery pumps there, the gas manifold flow reactor, and the online analysis. In this case, it's a GCMS, but we can do the same thing with HPLC mass spec. Now we have online NMR, FTIR, and Raman also integrated with our self-driving labs. And here's an example performance of the self-driving catalysis lab over time, showing that it can autonomously map the space of any given ligand for this homogeneous organic synthesis. And within 30 autonomously performed experiments, our self-driving lab could map the performance of this ligand for this given reaction. And if you want to do the same thing with a highly trained chemist, that would typically take you about 9 to 12 months. And then we did this for multiple different ligands, and then we transferred the knowledge discovered with this self-driving lab in a 0.5 mil flow reactor to a larger scale batch reactor with our industrial partner to show the knowledge of scalability that comes out of this self-driving lab. And we showed that if you use the same ligand that you discovered in your self-driving lab with the same reaction conditions and run in a larger scale batch reactor, you get the same selectivity and yield. And so that knowledge of scalability was very important for us. We spent about 1.5 years just to make sure that the knowledge we discover with this smallest scale reactor in a self-driving lab is scalable and directly transferable to our industrial partner. And then you can do a lot of things with the data that you generate with your self-driving lab. Here's an example of building a digital twin. This is a digital twin of the actual homogeneous catalytic reaction with different ligands. You can train a GP model, for example, for every ligand that you have tested, and then you can use that digital twin to visualize the reaction surface, to study the effect of different parameters on the yield and selectivity of your reactions. This is a great tool for better understanding of what's going on in the specific reaction you're studying. And then going back to the flow reactor, I mentioned that you can go from a single droplet to continuous flow. We basically use the optimized conditions to show that we can do this on-demand switching of selectivity of an organic reaction. There's a regio selectivity of the product. Using the conditions discovered by our self-driving lab, we could, on demand, on the fly, change the reaction conditions with the same ligand to change the regio selectivity of the aldehyde that we are producing in this flow reactor. And later on, we also use this method to show that we can also do hydroamine methylation in flow to produce alpha branch amines. And we showed that with our autonomous lab, we can basically discover the catalyst and even a co-catalyst here to accelerate the synthesis of hindered amines 70 times compared to the best performing batch reactions before. So those were just some examples of what you can do with a self-driving catalysis lab. But what is the future of autonomous labs? So what we're working on right now with our academic and national lab partners is on a vision of distributed autonomous labs that you can have this network of cloud-connected autonomous labs performing similar campaigns, sharing data over the cloud, and further accelerating discovery of new molecules, catalysts, and materials. But in order to do this and implement this vision, we need to work on the reproducibility, the knowledge transfer, and the data augmentation between different robots. Here's an example of the different language of different robots. Some might use a batch reactor, flow reactor, different types of even analytical techniques. So how do we augment this heterogeneous source of data and hardware? So we need to work on some level of abstraction. Too much abstraction, you're going to lose a lot of details. Too little abstraction is also not good. You have to do a lot of the definitions of different details of these autonomous operations. So that's something we're actively working on, and that is definitely needed to implement this distributed vision of these autonomous labs. The other important thing that we have to always think about is when we design a self-driving lab, we have to perform a lot of benchmarking tests, both on the hardware and the software side of these autonomous labs, to make sure that we can basically have this apple-to-apple comparison on the performance of these autonomous labs, from the chemical consumption to the in silico optimization models that they're performing. And then I'm going to end by, again, mentioning that we need to work on this knowledge transfer between different types of robots and autonomous labs. That's the only way we can really digitalize the discovery of new molecules, catalysts, and processes. If we cannot transfer knowledge and protocols, recipes, data, metadata between different labs, everything that we do will be only limited to our own in-house data that we are generating with our own robots. So at the end, I hope I was able to convince you that these autonomous labs are great robotic co-pilots to help us close the time and scale gap in molecular discovery, potentially 100 to 1,000 times compared to the conventional ways we typically approach molecular and catalyst discovery. Again, I want to thank all my group members who did all the work I presented today, and I want to thank our funding sources, my collaborators. I want to thank you all for listening. I'll be happy to take any questions.
Great, thank you so much. This is great presentation. It's very fascinating, self-driving lab. That's great topic. There is a couple of questions in the chat. Let me ask the first one. Yeah, that's interesting. How do you make sure what the self-driving lab does not turn into the Skynet?
Well, that's a question about the cybersecurity of these autonomous labs, and there's already a lot of activities and active programs working on the cyber infrastructure of these autonomous labs. Even within a university level, we have a lot of internal protocols on how to even remotely operate these autonomous labs, and then there are a lot of things you have to consider when you're working on industry-sponsored projects. How do you make sure that your data is completely secure, safe? What kind of servers you use? But the answer, I cannot answer it in the next 2 minutes, but the answer is a robust cyber infrastructure, and a lot of funding sources, federal funding sources in the U.S. are actually coming up with new programs on promoting the cyber infrastructure of autonomous experimentation and cloud labs, autonomous labs.
Great. The now question is, the microfluid approach is great for applying the chemistry reaction. How would you apply this to enzyme reactions or biological reactions for the drug discovery? Do you need to build different workflow to each different reaction?
That's a great question. So we don't start every single reaction in flow. We start, I briefly mentioned that we start with an initial parallel batch screening. So if you have a reaction that involves any sort of solid precipitation or a solid addition, we don't run it in flow. But if there is a reaction that we know that the reaction has no solubility problem, there's no precipitation, and there's no solid addition, we definitely run that reaction in flow. So we don't force feed reactions in flow, but when the reaction is all in solution phase, there's no precipitation challenge, and there's no solid addition during the reaction, and it's all solution phase, and we run the reaction in flow. Now, in terms of the design of the reactor, the design of flow reactors have evolved significantly in the last 20 years. Now, we are at a level that we can have these multi-purpose flow reactors that we can use for a variety of inorganic and organic synthesis without a need to redesign the workflow. They can use the same design, the same plates. And most of our flow reactors these days are tube-based, either Teflon tubing or stainless steel tubing, and it's the design of the heating plate that we typically change for different reactions. So the heating plate is reusable, and the tubes are very inexpensive to change.
And last question, and if you can answer some question in the chat would be great, but very last question, really quick one. So when we should expect the self-driving labs is going to be functional completely? Like, what is your forecast? It's like it's working today, we can run, or it's like 5 days from now, or 5 years from now? What is your like expectation?
Well,
what do you dream, what you want to design? Yeah.
The whole field of self-driving labs is still in the early stages, and there are a lot of different visions on how to evolve these autonomous labs. There's a vision that we just need human and robots, and that robot can go in the lab and just use the same the same glassware and tools that you have in an existing chemistry lab to do that. And there's the other vision that you need new tools, new hardware, new automation technologies to increase efficiency and reduce chemical consumption of your reactions. So I would say maybe there are a handful of fully autonomous labs, fully operational around the world. And it's going to probably take another decade to see going from handful to hundreds around the world. So we're still in the early days of these autonomous labs.
Got it. Thank you so much, Milad, for the great talk presentation.